Okay, Altica lythri is a flea beetle that feeds on antilobium, and it's known for its natural female bias. And our group has studied 14 populations across Germany, and the result shows, you can see on the figure, that it has a very strong female bias, as well as we have also found some reproductive anomalies that some individuals produce only female offsprings. The cause of the school-sex ratio could be very complicated. There are several sex manipulators that we suspect of. Firstly, is it could be caused by nuclear cytoplasmic conflicts, and that resulted from hybridization or other evolutionary events. And the second is some meiosis anomalies. Something could be happening wrong in meiosis. For example, genogenesis, that's a figure shown in the left. That means that the sperm only works as a trigger for the development of eggs, but the genetic material didn't transfer into the offspring. And the third one that we suspect of is bacteria endocrine bios. That's a very famous one. It's reported to infected over 60% of insects, and it could induce pathogenesis and male killing and other mechanisms that result in female bias. Wolbachia rickettsia is a very famous example in this category. And preliminary study has showed that we try to use single-locus markers to resolve the question, but nuclear markers, even the intro and RA genes, did not show a difference of normal reproduction and female bias reproduction.
I'm sorry to interrupt, Cen, but we do see the presenter view, meaning that your next slide as well. So if you can go to the full screen mode, then we can see your whole slide better, actually. Thank you very much.
Yeah. Is everything normal? Okay.
I think you have to share your screen with us again.
Okay. Sorry.
Perfect. Thank you very much.
Yeah. And yeah, where have I got? Okay, so the nuclear markers did not show the difference between normal reproduction and female bias reproduction, but we have used mitochondrial markers such as CO1 genes and NE5 genes that present the phylogeny on the right. It shows that it successfully shows distinct interspecific difference in genus Altica. And let me put, and our target group, Altica lythri, formed 3 different groups that has more p distance than interspecific differences. And these 3 haplotype groups has a p distance larger than 2%, and the haplotype HT1 is the group we're trying to find. It's the special group of female bias. And as our Altica lythri is broadly infected by Wolbachia, and I forgot to mention that we suspect that 2 out of 3 those mitochondrial haplotypes must come from hybridization with other species. And as it's broadly infected with Wolbachia, we want to ask, is this sex bias in Altica lythri related to Wolbachia? The evidence we have here is that usually one species only infected with 1 strain of Wolbachia because it's vertically transmitted from the mother. But in our case, we found three strains of Wolbachia infections. And And we found the linkage of mitochondrial haplotypes with 3 strains of Wolbachia, and especially female excessive haplotype HT1 that has a corresponding Wolbachia infection WA1, which produce only female offsprings. And that could also be evidence to support the assumption that mitochondrial DNA introgression accompanied by Wolbachia. But Wolbachia alone cannot explain the whole story, because we also found haplotype 2 and haplotype 3 with Wolbachia infections have rather balanced sex ratio, and the high frequency of uninfected females with haplotype 1 and 1 star cannot be explained by WA1 infection alone. What we know about the special type HT1 star is that no Wolbachia infection is detected, and it performs very similar with HT1 with only female offspring, but they have to mate to produce fertile eggs. And then we are trying to ask some questions about what's the cause of our school sex ratio. The first one, does the males contribute to the offspring, is presented by Alina this morning. And I'm focusing on the second and the third question is, are there nuclear exports of Wolbachia genes in unaffected HT1 females? And the third one is, are there sex or haplotype specific markers that cannot be found in HT1 or HT1 star make it so special? The method approach we are using is Pool-Seq and RAD-Seq, as well as we are also using the genome assembly and the transcriptome to perform this analysis. And Pool-Seq is called pool sequencing, and is to use DNA of multiple individuals from a population to sequence together. What we set up is 4 pools, including 50 individuals per pool, and we want to compare of HT1 and HT1 star for Wolbachia related markers, and HT2 female and males for sex specific markers. Here's a mapping result of our Pool-Seq reads on one of our contexts of our genome. We're trying to reach highly accurate genome-wide allele frequency estimates through those mappings. And we also want to get a large number of SNPs that we could perform as candidate of our target markers. The other method we are using is called RAD-Seq. It's a restriction site associated DNA sequencing. And compared to the traditional whole genome sequencing that showed in the left upper corner, the RAD is like a stable sampling of genome across multiple individuals, that they use the same set of restriction enzyme to cut through the genome, and then for similar individuals, it's not the same individual, but for like related individuals, we could get quite similar reads to form some stacks, and then we could call SNPs from those stacks, which produce very high accurate SNPs, while at the same time the Pool-Seq would produce more SNPs as it goes the whole genome wide, and we have more coverage on the genome. The RAD-Seq we're using is a modified one with 4 barcode system that increase the capacity of a single run, and that we could apply for more samples. Here is some part of my results. Is firstly that using the reads from ddRAD and the loci mapped to the Wolbachia genome in uninfected beetles, we found some of them has reads that mapped to the Wolbachia genome, which shows a potential export from the Wolbachia genome to our beetle. but it did not show a very comprehensive loci in every uninfected beetles. So the confidential level is not very high. We're trying to find more about that in our Pool-Seq dataset. And the second result we want to present is that we could see that in our ddRAD data, different haplotype poses more genetic variation than different sexes. We could see that for haplotype 3 and haplotype 2, they form different clusters by themselves instead of clustered by sexes. And the sexes that only possessed by female or males is missing in our case, so that our ddRAD data didn't reach such kind of region, which suggests a more highly likelihood that these two come from different hybridization of different species, but the origin is still unknown. And we also compare the data in population scale using a R package to perform structure-like plot. For example, for these two population Butian and population Weedle, they were separated for around 100 kilometers. It's quite far away for a nail-like beetle, but they share very similar genetic structure. But for this population Neuhaus, it shows that a quite distinct differentiated structure with the same haplotype from population Butian. And especially for haplotype 1 star, it shows more divergent than the haplotype 1. These two haplotype 1 star from different population is very different. And by Pool-Seq, we further searched the genome-wide differentiations. And in the y-axis, that present here, it shows minus log of p-value, which shows the confidence of the difference along this contact. And more close, this p-value is to 0. More likely, this site, this locus, is to be significantly different. And we could find like some area that has quite high differentiation level. But as we don't have a very perfect genome yet, our genome is very fragmented with more than 20,000 contacts. So we're still working on that. And we also applied the ddRAD data on other Altica species to study the origin of our Altica lythri hybridization. But currently, the species we are using didn't show a very close relationship with Altica lythri here. And the Treemix program shows very slight gene flow from Altica lythri to this branch, but the confidence level is not very high. We're going to add more Altica species to that, but the collection could be very difficult during the corona because of the corona. And here I couldn't draw very solid conclusion for my study, but I'm going to anyhow answering some of my questions. The first is, are there nuclear export from Wolbachia gene to uninfected HT1 females? We found that ddRAD and transcriptome data shows that the possibility of those exports, but we need to study more, especially on HT1 star, because it shows quite divergent types. And currently, we did not find individually different sex-specific and haplotype-specific marker in ddRAD data, but we
sorry to interrupt, but you have 1 minute left from your presentation time.
Yeah. And we still want to study more, especially in HT1 star haplotype. And we want to put further study on Pool-Seq data with Wolbachia related genes and sex-specific loci. And we also want to put more Altica species in our tree to find out the origin of our mitochondrial haplotype. And thank you very much for listening. And thank for the Altica project. And thanks for the project hybrids and the University of Hamburg. And thank you very much for listening.